Hello, I am Alexis Maya Connors. This is my second year teaching biology here. Before this, I was teaching at an elementary school for 2 years. I taught 4th grade science there. I graduated from the University of Connecticut with a bachelor's degree in early childhood education. After that, I went to grad school at Gallaudet, once again for ECE, early childhood education, along with deaf education, grades K-12. Read this and explain what haploid means. Read this and tell me. Those are gametes. Yes, but I'm asking you what haploid means. Read through the first sentence. A cell or organism. What does haploid mean? It means this, I don't know. Read it again and tell me. A cell or organism consisting of chromosomes. So, a haploid means, I don't know. Add up the number. Does it have enough or what? Gametes. Okay. "46. Now, how do you get 46 chromosomes in total? Yep, you have 2 parent cell. Now, how many chromosomes are from each parent cell? No. Separate 46 by 2. Yes, 23. Now, you see that there's 2 separate cells. What does a haploid mean? Explain it. Why was 46 split into 2 cells of 23? What does haploid mean? How is the 46 being halved? In the human body, there are a total of 46 chromosomes, right? Each parent cell gives 23 chromosomes and it becomes 46 in total, so that means each gives half. Do you understand what I'm saying? Now, what does haploid mean? Half. Here and here. How? Is it one cell? You get half from each parent cell. So, now we'll get into telophase 1. How many daughter cells are there? 2. Now, telophase? Does that cell have enough genes? How many daughter cells will there be by the end? 23. How many daughter cells? Yes. For meiosis, how many daughter cells are there? Not yet. Look. Telophase 1. How many daughter cells are there? How many daughter cells? Look. How many daughter cells? 2. Yes, but is that enough? Is that enough? It needs more. What's happening? It's halving again. Yes, it needs to have more daughter cells. A half. Yes. I'm going to ask again. What's in our body? For a baby, what do the parents provide it with? Yep, they make up the DNA. So, they halve their cells to make DNA for the baby. In metaphase 1, are there tetrad or do sister chromatids conflict? Yes. So we're finished combining the parent cells. This is during prophase 1. What does this mean? Swap. The parent cells swap their genetic material. What's this? What's this called? Yep. And what's this called? Yep. It means the chromosomes swap their genetic material. Yes, sex cells. Or there's another name. Yep. Gametes. How? During meiosis 1, are there sister chromatids or individual tetrads? They pair up. Now, what happens during meiosis 2? Are they still together or what? They separate. So what does that mean? Yes, the sister chromatids are separated. They become single. No, they don't swap. Yes, that means all the chromosomes have to match. They have tetrads. How? What are you doing? What did you say? Yes, then, so they all connected or separated? Yes, connected. Now let's move on to prophase 2. What happens? They separate, which means all the individual tetrads have the same genetic material from one parent. What does the separation do? From here to here, what happened? They swapped genetic material. I finished explaining what happens during prophase 1. Let's move on to what happens during metaphase 1. So, it's all the same vocabulary. Add it to the list. What's this? What's this? They're connected, they half, yes, and then the haploid. No, not the cell. Remember, how many chromosomes do our bodies have? Yes, how do you get to 46? Who does the cell get it from? Who does it come from? Who gives the genetic material? Yes, the two parent cells. How many chromosomes do they each give? 46 divided by 2. Yeah. 9.8? Who is involved in the production? Where do you get your genetics from? Yes, so what does it do with the genetics swapped from each cell? Yes, they come together and it splits it in half. So does that mean that genetics are all the same or different? How could genetics be different? What happens in prophase 1? These two sets of 4 chromosomes are called what? It's 2 words. I feel you. I have it here. What's there? Yes. We have 4 minutes. If you're not finished, you can come here after school and finish your project. You're separate. Okay? I'll explain what we're doing with the project tomorrow. You can come talk to me after school. That's last. That's the last thing you turn in. Okay? It's due tomorrow on Tuesday because Wednesday. There's no school on Friday and you don't have class with me on Thursday. Follow the odd schedule. No. Follow the odd schedule. Okay, so what is my structure? First, I use the 8 methods of both languages. For example, before there's a preview, then view, then a review. Often, science is viewed as a college concept, meaning it has a ton of vocabulary. So I review it again and again so it sticks in their brains before they forget it all, and I relate the concept to their current life. Then I also do a lot of hands-on building and lab activities. They have to understand the vocabulary first and connect it to the concept itself. And finally, I challenge them, either with writing or signing, using their vocabulary to explain what things mean, its process, what it's made of. For example, I have to teach mitosis. The students have to understand what the process is, what happens in each stage. First, they explain the vocabulary, the concept of cells dividing, and what's happening during that process. There's a lot of PVR, preview, view, review. They have to learn this every semester so it sticks to them. Most deaf or hard of hearing students have a hard time retaining the vocabulary, so I teach it time and time again so they can retain the lesson, and I have to challenge them.